Imagine if the history of an entire continent had been hiding inside something smaller than a grain of dust. For centuries, scientists, archaeologists, and historians have argued over one enormous question. Where did the first peoples of the Americas actually come from? Was there one migration or several? Did everyone pass through the same ancient gateway, or were there hidden branches that disappeared without leaving a trace? And why do some indigenous American populations carry genetic signals that seem connected to people living thousands of miles across the Pacific? For a long time, every new discovery seemed to create another mystery. But now, ancient DNA has changed the picture again. In 2026, one of the largest and most geographically diverse genomic studies of indigenous Americans ever conducted revealed an astonishingly complex history of migrations, isolation, survival, adaptation, and ancient encounters. The mystery has not simply been answered, it has been transformed. For generations, the story seemed almost simple. Modern humans evolved in Africa, spread across Asia, and eventually reached the far northeastern edge of the continent. During the last ice age, enormous amounts of Earth's water were locked inside glaciers, causing sea levels to fall dramatically. Where the Bering Strait exists today, a vast region of exposed land connected Siberia and Alaska. Scientists call this ancient landscape Beringia. It was not simply a narrow bridge that people crossed in one afternoon. It was a huge ecological region containing rivers, grasslands, animals, and changing climates, and humans could have lived there for thousands of years. Somewhere within this enormous northern landscape, populations related to the ancestors of indigenous Americans became separated from other East Asian populations. Over time, genetic differences accumulated. Eventually, some of these people expanded into the Americas. But the deeper scientists looked, the less this appeared to be a single journey with a single starting point. One of the most important clues came from ancient DNA. In 2018, researchers analyzed the genome of an infant known as Upward Sun River 1, who lived in Alaska around 1,500 years ago. The child belonged to a population scientists described as ancient Beringians. The genome helped demonstrate that the ancestors of indigenous Americans had a complicated history in Northeast Asia and Beringia before populations spread throughout the Americas. Earlier genomic work had already suggested a period of separation between ancestral indigenous Americans and other East Asian populations, with later movement through Beringia. The significance was enormous because archaeology could show where ancient people lived, but DNA could begin to reveal how those people were related. Suddenly, the ancient landscape was no longer just a map of archaeological sites. It became a genetic family tree, although even that phrase is almost too simple for what the evidence eventually revealed. The old picture had another major problem. For much of the 20th century, the Clovis culture occupied a special position in discussions about the first Americans. Clovis sites, recognized by their distinctive stone tools and associated archaeological remains, were traditionally dated to roughly 13,000 to 12,600 years ago. Because these sites were among the earliest widely recognized archaeological evidence in North America, some researchers once imagined that the people associated with Clovis might represent the first major population to spread across the continent. But as archaeologists discovered evidence of human presence that appeared older than or independent of Clovis, that idea became increasingly difficult to maintain. The Americas were beginning to look inhabited before the classic Clovis horizon emerged. The question was no longer simply whether Clovis represented the first Americans. It was how Clovis fitted into a much older and more complicated population history. Then came one of the most famous ancient DNA discoveries in the debate. In Montana, researchers recovered the remains of a young child from a Clovis-associated burial. The individual, known as Anzik I, lived approximately 12,600 years ago. When scientists sequenced the genome, they found something that had enormous implications. Anzik 1 was genetically connected to indigenous American populations rather than representing evidence for a European origin of Clovis people. The result provided strong genetic evidence against the idea that the Clovis population had originated from Ice Age Europeans. The DNA did something archaeology alone could not do. It placed a specific ancient individual into a genetic relationship with living populations. The mysterious people behind the tools were no longer anonymous. Their genome was telling a story of ancestry that pointed toward the indigenous peoples of the Americas. But this is where the story becomes much more interesting. Every time scientists thought they were approaching a simple answer, another layer appeared. Anzik 1 did not prove that every ancient person in every region of the Americas belonged to exactly the same population. Instead, the expanding ancient DNA record began showing that the first settlement of the Americas involved population branches, movements, regional differentiation, and later episodes of contact. Some ancient groups left substantial ancestry in later populations. Others appear to have contributed little or no detectable ancestry to people living today. Some populations remained genetically stable for thousands of years. Others moved, mixed, or were replaced. The continent was not populated by a single wave that simply marched southward and filled an empty map. It was a dynamic landscape of human movement. And this is why the newest research is so important. On April 22, 2026, a major study published in Nature presented data from 128 high-coverage Indigenous American genomes. The genomes represented 45 indigenous populations, 28 language families, and communities across 8 Latin American countries. This was not simply another handful of ancient skeletons. It was a major attempt to expand genomic representation of indigenous American populations, many of whom had historically been underrepresented in global genetic datasets. The researchers found extensive genetic diversity that had previously been poorly characterized. Their analysis indicated at least 3 major dispersals into South America, followed by regional differentiation and long-term genetic continuity. In other words, the newest evidence did not erase the older Beringia story. It added multiple chapters after that initial movement. The first great lesson is therefore surprisingly simple. The ancestors of indigenous Americans were not a single genetically uniform group. They were populations whose histories diverged over time. Imagine looking at a river from above. At its source there may be one channel. Then the water divides into branches, some of which reunite, some of which disappear, and some of which flow into completely different landscapes. Human populations can behave in a similar way. A founding population can separate into groups, those groups can migrate into different environments, and thousands of years later, their descendants can look genetically distinct. Then another migration can introduce new ancestry into one region without transforming another. That is much closer to what the genomic evidence now suggests happened across the Americas. The 2026 study found evidence for at least 3 major dispersal events into South America. This is particularly important because South America was once treated in simplified models as the final destination of a relatively straightforward expansion. The new data instead indicate repeated population movements and regional restructuring. One of these later dispersals appears to have involved populations related to Mesoamerica, and probably occurred at least around 1,300 years ago. Scientists emphasize that this should not be imagined as a single invasion or a single historical event. Genetic evidence cannot tell us that a particular army crossed a particular border on a particular day. It points to patterns of gene flow and population movement over time. The real story is likely more gradual, involving increasing connectivity between regions, including possible connections involving Mesoamerica, the Caribbean, and South America. That detail changes the meaning of the word migration. When people hear migration, they often imagine one population packing everything it owns and moving from one place to another. Ancient DNA frequently tells a different story. Migration can happen in pulses. It can happen through small groups. It can happen over centuries. A few families can move into a new region, marry local people, and leave a genetic signal that survives long after their cultural identity changes. Trade networks can increase contact without replacing populations. Climate shifts can encourage movement. New technologies can spread without large-scale genetic migration. And sometimes a population can move while its descendants later become almost impossible to distinguish from neighboring groups. The genome records the biological consequences of these processes, not every detail of the human experience behind them. One of the most remarkable findings concerns a genetic signal associated with present-day Australasian populations. Several Indigenous American groups, including populations in South America, such as the Awajun, Ayoreo, Guarani, Caritiana, Siriano, Surui, and Simene, show an unusual genetic affinity with present-day Australasians when compared with other populations. At first glance, this sounds almost impossible. Australia and South America are separated by an enormous Pacific Ocean. There is no evidence that a population of modern Australians simply sailed across the Pacific and founded South American societies. The genetic signal requires a much more ancient explanation. The 2026 study points toward an unsampled ancient Asian population that researchers call Ipiquera, a name derived from an indigenous Tupi language and meaning ancestor. This population is not represented by a skeleton that scientists can simply place in a museum. Instead, its existence is inferred from patterns in DNA. Think of it as a missing branch in a family tree. You cannot see the person directly, but you can recognize that two visible branches share genetic features that make sense if an ancient, now unsampled population existed somewhere deeper in their history. Ipiquera appears to have been related to the ancestry of present-day Australasians and also contributed ancestry to some populations ancestral to Indigenous Americans. The amount of this ancestry in present-day Indigenous American populations is small, generally estimated around 1 to 3%, but its importance is far greater than its percentage might suggest. A tiny genetic contribution can preserve evidence of an ancient encounter that happened more than 10,000 years ago. The DNA does not mean that an Australian population suddenly appeared in South America. It suggests that the ancestors of these populations had already interacted somewhere in ancient Asia before the Americas were fully populated. The exact geographic location and route remain uncertain, and that uncertainty is crucial. The discovery is not proof of a direct trans-Pacific voyage by Australians. It is evidence for a much older and more complicated population history in Asia. This is where the phrase ghost population can be useful, as long as we understand what it means. A ghost population is not a mysterious civilization that vanished overnight. It is a population inferred from genetic evidence that has not yet been directly sampled or identified through a known ancient genome. Scientists have used similar concepts in many areas of human evolution. DNA can reveal that two groups share ancestry from a population that has not yet been discovered archaeologically or genetically. Ipiquera is therefore not a fantasy lost civilization. It is a scientific inference generated by genetic patterns. One day, researchers may find ancient remains that clarify who these people were, where they lived, and how they interacted with other populations. Until then, the genome provides the clue. And there is another reason this finding matters. The Australasian-related signal appears to have survived at low levels for more than 10,000 years. The 2026 research suggests that some genomic regions associated with this ancestry may have been influenced by natural selection. That means the story may not simply be about an ancient meeting between populations. Some inherited variants may have remained in populations because they interacted with environmental pressures or biological processes. Researchers identified signals involving genes associated with metabolism, immunity, reproduction, and development. This does not mean scientists can look at one gene and determine exactly how an ancient person lived. Natural selection is complicated, but it shows that the genetic history of indigenous Americans was also an evolutionary history. Imagine those first populations moving through completely different environments. Some encountered Arctic conditions. Others entered forests, mountains, grasslands, deserts, coastlines, and tropical regions. Food availability changed, pathogens changed, temperature changed, altitude changed, daylight patterns changed. The pressures affecting survival were not identical everywhere. Over thousands of years, natural selection could act on genetic variation already present in populations or introduced through migration and gene flow. The result was not one universal indigenous American genome. It was a mosaic of populations shaped by different environments and different histories. The new research identified candidate genes connected to immune responses and other biological functions, including CMKLR1, CCDC134, and DOCK2. It also found signals in regions involving genes such as FADs, TOIS, FADS3, and SLC-TOM5A17. These findings do not mean that a particular gene has one simple function that explains an entire population. Human traits are usually influenced by many genes and environmental factors. But collectively, these patterns show that indigenous American populations experienced evolutionary pressures over long periods of time. Their genomes preserve evidence of adaptation to the environments in which their ancestors lived. Yet perhaps the most important lesson from the latest research is not a particular gene or migration route. It is continuity. For a long time, ancient populations were often treated as if they were isolated snapshots. A skeleton would be discovered, dated, and described, and scientists would ask where that individual fit in the larger story. Modern genomics allows researchers to connect multiple snapshots across thousands of years, and sometimes the result is extraordinary continuity. A 2025 Nature study focused on central Argentina and analyzed genome-wide data from 238 ancient individuals spanning roughly 10,000 years. The research identified a previously unsampled deep lineage whose earliest representative dates to around 8,500 years ago. In central Argentina, ancestry associated with this lineage persisted for thousands of years with relatively little evidence of interregional migration. Later, that ancestry became involved in several different gene flow events, including movement into the Pampas and connections with populations in the Andes and the Gran Chaco. This is a remarkable example of a population lineage surviving through immense stretches of time while still participating in later demographic changes. Now travel thousands of miles north and look at Colombia. In 2025, researchers published a genomic transect from the Bogotá Altiplano, covering approximately 6,000 years of human history. They analyzed 21 individuals from archaeological sites dating between roughly 6,000 and 500 years ago. The results revealed multiple genetic shifts through time. Early hunter-gatherers in the region did not simply fit into the genetic categories scientists had expected. Their ancestry reflected an ancient lineage connected to the broader early population history of South America, but they also showed a distinctive genetic profile that did not map neatly onto later populations. Then, around 2,000 years ago, major genetic changes appeared, consistent with new population movements and gene flow into the region. That Colombian evidence is incredibly important because it demonstrates something the broader continental story keeps repeating. Population history can change without leaving behind a simple archaeological boundary. A new type of pottery does not automatically mean a new population arrived. A new language does not necessarily equal mass migration. A new agricultural practice can spread through cultural transmission. Conversely, a genetic shift can indicate population movement even when archaeology alone cannot identify the event. Ancient DNA allows scientists to separate these processes more carefully. It can reveal moments when culture changed, but ancestry remained stable, and moments when ancestry changed, even when the archaeological record looks less dramatic. This distinction matters because the Americas were never culturally or genetically frozen. Populations traded, moved, intermarried, separated, adapted, and sometimes disappeared. Some lineages expanded dramatically, others became geographically restricted. Some appear to have vanished genetically, leaving no obvious modern descendants. This does not mean their cultures or achievements were insignificant. Genetic disappearance is not the same thing as cultural disappearance, and genetic continuity is not proof of cultural continuity. Human history cannot be reduced to DNA alone. But DNA gives us another layer of evidence that can reveal demographic events that were invisible before ancient genomes became available. And then there is the question that has fascinated scientists for decades. How exactly did the first people enter the Americas? The strongest evidence still points toward an ancestral population related to Northeast Asians and movement through Beringia. But the timing, routes, population structure, and sequence of events are much more complicated than a single arrow on a map. There may have been periods of isolation in Beringia. There may have been multiple movements southward. Different branches may have entered or expanded through different pathways. Some groups may have moved rapidly along the Pacific coast. Others may have followed inland routes as environments changed. The archaeological and genetic records are still being combined to reconstruct the details. What the latest research does not support is the idea that the entire indigenous American population originated from a mysterious European migration that replaced an earlier population. The genetic evidence accumulated over decades points overwhelmingly toward deep ancestry connected to ancient populations of Northeast Asia and Beringia. The famous Clovis-associated Anzik 1 genome fits within the ancestry of indigenous Americans. Ancient genomes across the Americas show continuity and relatedness patterns that make a simple European founding population unnecessary. Alternative hypotheses have repeatedly been tested against genomic evidence, and many do not survive that comparison. But the opposite extreme is also wrong. The scientific story is not that every indigenous American population can be traced through one perfectly straight genetic line back to one small group in Siberia. The 2026 evidence makes that simplification even harder to defend. The Americas were populated through a complex sequence of demographic processes. South America itself experienced multiple dispersals. Later movements connected distant regions. Some populations carried unusual ancestry from ancient Asian sources. Other lineages persisted locally for thousands of years. Some disappeared. Others became major contributors to later populations. So when we say scientists have finally solved the Native American DNA mystery, what does solved actually mean? It does not mean every question has an answer. It means that the central framework has become dramatically clearer. We can now see that indigenous American history is not one unexplained event, but a long sequence of population histories. We have stronger evidence for the Northeast Asian and Beringian roots of the founding populations. We have ancient genomes that connect early individuals to later indigenous Americans. We have evidence for multiple dispersals within South America. We have detected ancestry from an ancient unsampled Asian population. We have found evidence of long-term regional continuity and natural selection. And we have learned that the genetic diversity of indigenous Americans has been profoundly underrepresented in global genomic research. That last point is especially important. For decades, genetic databases were disproportionately built from populations of European ancestry. That imbalance affected what scientists could see. If a population is poorly represented in genomic datasets, its unique variants and evolutionary history remain hidden. The 2026 Indigenous American Genomic Diversity Project directly addresses that problem by generating high-coverage genomes from a broad range of indigenous populations and working with communities. The goal is not simply to collect DNA and publish statistics, it is to improve understanding of human diversity while recognizing that indigenous communities should have a meaningful role in how their genetic information is studied and interpreted. The result is already changing the map of human genetic diversity. The 128 new high-coverage genomes revealed extensive variation that had not been adequately characterized. When those genomes were analyzed alongside previously published data, researchers could identify patterns that were invisible when the data set was smaller. This is a fundamental principle of population genetics. The more diverse the sample, the more accurately scientists can distinguish ancient population structure from recent demographic events. A pattern that looks mysterious in 10 genomes may become obvious in 100. Another pattern that appears simple in 100 can become complicated when more populations are included. And that is exactly what happened here. More data did not make the story simpler, it made it more accurate. The same principle explains why the word ghost keeps appearing in ancient DNA research. Every new genome is another window into the past, but it can also reveal that the people we have already sampled are only fragments of a much larger population network. If a genetic signature appears in two distant groups, but not in the populations we have sampled between them, scientists must consider the possibility that an unsampled ancestral population carried that signature. The missing population may have lived somewhere in Asia, interacted with the ancestors of multiple groups, and then disappeared as a distinct population. Its DNA could survive even when its name, language, and archaeological identity are lost. This is one of the strangest things about DNA. A person can disappear from history without disappearing completely. A population can vanish as a recognizable community, while fragments of its genetic inheritance continue inside other populations. A mutation that began in one ancient individual can travel through generations and eventually appear in people living thousands of miles away. A population can migrate, mix, and transform while preserving tiny genetic traces of earlier encounters. DNA is therefore not a complete autobiography. It is more like a collection of footprints. Some are enormous and obvious, others are so faint that scientists need statistical methods to recognize them. But together, those footprints can reconstruct journeys that happened long before writing existed. And the journey of Indigenous Americans is one of the clearest examples of this principle. Their ancestors survived dramatic climatic changes, entered unfamiliar ecosystems, established communities across enormous distances, and developed adaptations to environments ranging from the Arctic to the Amazon. Their descendants maintained regional genetic continuity for thousands of years, while also participating in migrations and exchanges that reshaped the continent. The genetic evidence shows both movement and stability, both isolation and connection. There is also a human dimension to this story that numbers cannot capture. When researchers describe a genome as belonging to a population that lived 8,500 years ago, they are not describing an abstract data point. They are looking at the biological remains of an actual person who lived through a world completely different from ours. That person ate food, experienced weather, formed relationships, learned skills, and lived within a community. Their DNA survived because the molecules inside their cells preserved fragments of a biological history that continued through generations. Modern sequencing technology can read those fragments, but it cannot tell us everything that made that person's life meaningful. That is why ancient DNA should not be treated as a replacement for archaeology, anthropology, oral history, or indigenous knowledge. It is another form of evidence. The strongest reconstruction comes when different kinds of evidence are brought together. Archaeology tells us about objects, settlements, technologies, and behavior. Linguistics can reveal relationships and historical changes among languages. Environmental science can reconstruct climates and landscapes. Anthropology provides cultural context. Oral traditions preserve knowledge through generations. Genetics can reveal biological relationships and demographic movements. When these lines of evidence converge, the picture becomes much stronger, and sometimes they disagree, which is equally valuable. A disagreement between archaeological and genetic evidence is not necessarily a failure. It can indicate that scientists have misunderstood a migration, overlooked a population, or assumed that cultural change and genetic change happened together. The 2025 Columbian research is a good example of why this matters. Genetic evidence revealed demographic shifts that could not simply be explained by assuming that every archaeological change represented the arrival of an entirely new population. Human societies are more complicated than that. The deeper we go, the more the old image of the first Americans changes. They were not one mysterious group that appeared suddenly. They were part of a much longer Asian population history. Their ancestors experienced separation and movement. Their descendants spread across enormous territories. Some branches remained connected, others became isolated. Later populations moved through the continent. Some genetic lineages persisted for thousands of years. Others left little detectable ancestry today. Ancient encounters left tiny traces. Natural selection shaped the genomes of people living in different environments, and all of these processes unfolded over a time scale so vast that human historical records cover only a tiny fraction of it. The title of this story might say that scientists finally solved the mystery, but perhaps the most surprising conclusion is that the real mystery was never a single question. It was a network of questions hidden inside one another. Where did the first people come from? How many groups entered the Americas? How long did they remain in Beringia? Which populations moved south? Which ones disappeared? Which ones survived? Why do some South American groups show Australasian-related ancestry? Where did Ipiquara live? How did its ancestry reach the ancestors of Indigenous Americans? Why did certain genetic variants survive for thousands of years? Which migrations reshaped South America after the first settlement? And how much of this history is still invisible because the relevant ancient genomes have not yet been discovered? The 2026 research gives us stronger answers to some of these questions, but it also tells us where the next discoveries are likely to come from. Scientists need more ancient genomes from regions that remain poorly sampled. They need better geographic coverage across northern Asia, Beringia, Central America, the Caribbean, and South America. They need more collaboration with indigenous communities. They need improved statistical models that can distinguish ancient population structure from later gene flow. And they need archaeological discoveries that can be matched to genetic signals. The next major breakthrough may not come from finding a famous skeleton. It could come from one small fragment of bone discovered in a location that scientists have barely sampled. That fragment could change the family tree again, because the history written into DNA is not finished. Every new genome can move a branch. Every new archaeological site can change the timeline. Every new population sampled can reveal genetic diversity that scientists did not know existed. And every ancient individual carries a combination of ancestry that may connect distant regions in ways no historian could have predicted. Think back to the question we started with. Where did the first Americans come from? The answer is no longer simply Asia. That is true, but it is incomplete. The deeper answer is that the ancestors of Indigenous Americans emerged from ancient populations in Northeast Asia, experienced a period of differentiation associated with Beringia, and then participated in a long sequence of movements and population changes across the Americas. South America was reached through complex dispersals. Later migrations reshaped individual regions. Ancient Asian ancestry that has not yet been directly identified left a small but persistent genetic signal in some indigenous populations. And after thousands of years, the descendants of these populations still carry evidence of the journeys their ancestors made. You are carrying the history of migrations. You're carrying the history of survival. You're carrying traces of people who lived before written history, before cities, before borders, and before anyone had a word for genetics. And that may be the most astonishing discovery of all, because DNA does not remember history the way a book does. It does not preserve names, dates, speeches, or monuments. Instead, it preserves fragments, a small change here, a shared sequence there, a genetic branch that appears in one population and disappears in another, a tiny percentage of ancestry that should not be there unless something happened thousands of years ago. Scientists take those fragments and reconstruct the shape of a human story. For the first peoples of the Americas, that story is becoming clearer than ever before. But it is also becoming more human. The old image was a map with one arrow pointing from Siberia into America. The new image looks more like a web. Branches spread, some merge, some vanish, some remain isolated for thousands of years. Some carry traces of populations that scientists have never directly found. Some adapt to mountains, others to forests, deserts, coastlines, or frozen northern landscapes, and through all of it, people survive. That is what the newest DNA evidence really reveals. The mystery was never that indigenous Americans had one secret origin that scientists simply needed to uncover. The mystery was that an entire continent preserved a record of human movement far more complicated than anyone had imagined. And now, after decades of archaeology, genetics, ancient DNA sequencing, and increasingly diverse genomic research, we can finally see much more of that record. Not the whole story, not yet, but enough to understand something extraordinary. The first Americans were not the end of a journey. They were the beginning of thousands of journeys. Their descendants carried those journeys forward, generation after generation, until fragments of them reached the present. So the next time you look at a map of North or South America, remember that the borders printed on it are incredibly recent. The human map underneath them is ancient. It was drawn by movement, separation, survival, adaptation, and connection. It was written long before anyone could record a single word. And some of it is still being written today, not in ink, not in stone, but inside us.